Thanks, Jason. It's my pleasure to be here today. I have the unenviable position here of speaking right after lunch, so I'll try not to be too loud or annoy anybody too much here. I thought I'd get started just giving a little bit of background about the Food Research Institute. I'm sure some of you in the audience are familiar with it, but some of you may not be. We've been around for quite a while. We're in our 73rd year. It was actually started by a physician, Dr. Gail Dack at the University of Chicago. And following his retirement in 1966, he moved up to the University of Wisconsin, where it's been ever since. It was pretty simple back in the days of Dr. Dack. You were worried about salmonella, botulism, staph enterotoxin, a couple other things. Now we have a long list and a growing list, it seems at times, of foodborne pathogens that we're concerned about. Within the group, we currently have 24 faculty and staff in 8 different departments in 3 different schools and colleges. So we're interdepartmental, interdisciplinary, and everybody in the group has something about their research efforts that focuses on food safety. We cover all the major bacterial pathogens with particular expertise with Clostridium botulinum and its toxins, and also we're quite strong with fungi and mycotoxins as well. And what enables us to accomplish our mission is the support we receive from sponsor companies. And I suppose this is my disclosure then that there's a number of companies I have listed here, and representatives of some of these are in the room here today, who are sponsors of the mission of the Food Research Institute, provide unrestricted gifts to us to support our staff and the various projects that we undertake. None of the work I'll be talking about today is directly supported by any funding from any of these companies, but we greatly appreciate the relationship we have with them and try to help them deal with the food safety problems that they grapple with on a daily basis. And in particular, we have the Applied Food Safety Laboratory run by Dr. Kathy Glass. Anybody who works in the realm of food safety knows Kathy, and she just has an exceptional staff who can run food challenge studies with whatever problem or type of food product you're concerned about. I'll also mention an exciting addition to campus that's going to impact our program and offer the opportunity to do even more. There's a new meat safety and biological discovery laboratory going up. It should be online this fall. This will be a world-class facility for training students in meat biology, meat science, and we'll also have a BSL-2 lab attached to it, separate air handling and water system that will allow us to do challenge studies either on an intact animal going in for slaughter or a challenge study with the resulting product you might make from that animal. So it'd be an exciting possibility. I'll also put up a brief ad here for an outreach effort that we're conducting this fall. Every year we work with the Institute for Food Safety and Health, the group that Bob Brackett runs, that's a consortium of the FDA and Illinois Institute of Technology, and we co-sponsor a symposium in Chicago each year. This year, the topic is managing microbiological testing as a preventive control verification. It'll be held in the Chicago suburbs, October 24th. Program's set and online, registration's open. If you have any questions, I'd be happy to answer it and hope to see some of you there. So I was asked to talk a little bit about the sorts of things that my lab does, and I'm a scientific generalist. I've probably never seen a project I didn't like. It's my disease, and been involved in a number of microbiological projects over the years, and I'm listing some of the current ones up here. For a long time, we've worked on bovine respiratory disease, some of the bacterial pathogens there. Not going to talk about that today. And I'm also part of a large group that have been using nanotechnology and other efforts to develop novel dressings to improve wound healing and reduce biofilm formation within wounds. I'm not going to talk about that either. But we have a long-standing interest with foodborne pathogens, particularly Listeria, and also to a lesser extent with Salmonella, or a more recent extent with Salmonella. I'm going to talk a little bit about some of the more applied projects that we've been involved in recently. And these are basically surveys of the microbiome, or perhaps it'd be more accurate to say bacteriome, and certain environmental conditions that might influence the ability of pathogens like Salmonella and Listeria to survive and perhaps cause problems in animals and people. And I'll talk a bit about work done by Hannah Pilch, who's in the lower right-hand corner there, who did an analysis of the microbiome of bedding sand used for dairy cattle. I'll tell you why that's relevant and how that might support the survival of Salmonella in that environment. I'll also talk a little bit about work done by Kurti Wadawan, who's at the bottom of the slide there, who's been looking at the microbiome of wooden boards used for aging cheese. This is obviously two disparate topics here, so I'm going to try to link these together a little bit in terms of the overall interests we have. And we can only accomplish this work with the collaboration with our colleague Garrett Soon in the upper right-hand corner. Garrett's an outstanding scientist studying the microbiome and has been really helpful to us in conducting these studies, and I'm quite grateful for the help he's given us. And I might mention Hannah recently defended her PhD and as of yesterday is now an employee of Carrier Ingredients, so happy to see her move on in life. So I'll talk a bit about the work we've done with bedding sand, and the underlying rationale here is the concerns we have with Salmonella as a foodborne pathogen. I really don't have to tell you too much more about this. It's either number one or number two amongst the bacterial pathogens on an annual basis in the United States, probably causing 1 million or more cases of foodborne disease. Some of these can be quite serious and even lethal. And most of these are contracted by ingestion of contaminated food products, but you can also, human cases will also occur from direct contact with infected animals or the organism being shed into the environment by various animal species. And in particular, being in Wisconsin, we're concerned about dairy cattle and the dairy environment. And Salmonella is of concern in dairy cattle, both as a pathogen, particularly in young animals. But we also have a situation here where the older animals can become colonized with the organism, periodically shed it in feces. And in particular, they can become colonized in their lymph nodes with little or no apparent adverse effect on the animal in many cases, but they're still then a source of the infection for other animals, for the environment, or for people. And we also know Salmonella is an impressive organism in terms of ability to survive in the environment. Various published reports of it surviving 60 days or more in cow manure. That's probably an underestimate in my opinion. Over 150 days in water, et cetera. And I'll be talking a little bit about this ability in a minute. But as you can see here by the data on top here, this is fairly commonly distributed amongst dairy herds. It will vary from one region to another. But some studies done in Wisconsin estimate that about 30% of the dairies, and it's probably higher than that, have one or more animals infected with Salmonella. And when dairy cows go to slaughter, because of poor performance, cull cows, a good proportion of those will carry Salmonella as well. So one of the reasons why this is a concern is this lymph node colonization. This can occur in various peripheral lymph nodes in the bovine. And again, reports differ in terms of what the prevalence is, how common it is, and probably depends on breed and region and varies from farm to farm. But this is a substantial proportion of animals that will have this colonization. And there are certain serotypes of Salmonella. As you probably know, there's thousands of serotypes, but some of these seem to be quite adapted to the bovine. And it's some of these that you'd find most commonly then associated with lymph node colonization. And some of these isolates will be antibiotic resistant. So if they do get into the human population and cause infection, you can have some more difficult situations to deal with. And we've had some examples of that. Wisconsin recently, there was actually the Midwest, there was a big outbreak with Salmonella Heidelberg that was traced back to some dairy calves, et cetera. So we've had some pretty interesting and substantial outbreaks from time to time. So the major reason this lymph node colonization is concerned from the human health standpoint is that every dairy cow has a second career as a meat animal. You'll milk a dairy cow for a period of time, and eventually then that animal is not of economic use to the farmer. There's no retirement home for old dairy cows. They'll go to slaughter, and many of these will end up in ground beef that if you're eating a burger or whatever, that might be where it comes from. So when the animal goes to slaughter, you'll remove the prime cuts, the steaks, the roast. The rest of it, the beef trim, then will be ground up to produce ground beef. And it's likely these lymph nodes will be in that beef trim. And so if they contain Salmonella, the Salmonella is going to end up in this ground beef. And there have been some outbreaks related to this. This is an area of concern. And we'd like to get a better handle on it and understand how is the organism getting into these animals, how is it being maintained in the environment. So we decided to focus a bit on the way you bed dairy cattle just to see what organisms are present and then to ask questions related to the ability of Salmonella to survive in that environment. Now, based on the talks this morning about space and space exploration, I should have had a cow in a space suit. I actually found a couple online. I missed an opportunity here, but to focus in on what you often, if you have dairy cattle in a confined environment, and typically you would, you have to provide some kind of bedding for them. And there's all sorts of things that can be used, but sand has proven to be quite popular, and for good reason, particularly in the upper Midwest. It's good for cow comfort. They're comfortable. They have less lameness. It's cleaner than some of the other things you might use. A happy cow gives you more milk, leads to a happy farmer. And so bedding, sand for bedding is commonly used in this situation. Well, that's fine, but this sand is not going to stay pristine. What are you going to do with this in your situation on the farm? And what many places will do, they won't take all the sand out and bring fresh sand in. They will have some kind of a recycling process. And it's probably a little hard for you to see that from where you're at, and I don't know if I can use a pointer or not. But basically, we have animals in stalls. You'll take the sand, push it out into some kind of central alleyway, get it into some kind of a water flowage system. Ultimately, it flows into some kind of a lagoon. The sand is dense, it will sink. Everything else is less dense, will float. That'll be removed and composted or eliminated from the system. Then they'll have some way of sucking the sand out of the bottom of this pool, bring it up through an auger system, and during this process, they'll spray it with a disinfectant. And so the farm we've worked with actually uses hypochlorous acid for this purpose. Now, this is not going to sterilize this sand, as you might expect. There's no way you're going to get enough of it in there, but it does make some effort to control pathogens that might be present. And then what you do then is the sand gets dumped out of this system. They'll usually, when we're talking about larger operations that we do it in this way, drop it into large piles. It would sit there to dry out a little bit for a period of time, up to about 2 weeks or so. And then ultimately it's moved back into the stalls and the cows are put back on it. So this process does two things. One, it saves the farmer money, and also it's a sustainability effort. Instead of bringing fresh sand in each time, which you have to mine from somewhere, you can reuse about 90% of it. These systems are pretty good at recapturing the sand. But the question we had is, well, okay, this sand obviously has been in contact with the animals, with their manure. There's going to be a microbial community in there. What is there? And there have been previous studies of microbes that are in bedding sand, but these have used classic culture-based methods. Together with Garrett, we decided to take more of a molecular approach and try to characterize the bacteriome, if you will, of the sand. So what organisms are present? Does this change as you go through the recycling process? Is there a seasonal effect? And do we see carriage of pathogens throughout this process? And so these were some of the questions we were interested in answering. So the work here is done by Hannah. This was done on one dairy farm, a Jersey dairy farm in Wisconsin. It would be considered a CAFO. They had about 1,800 dairy cows. Very nice operation, very clean, very modern, excellent ventilation, everything else for the animals. What Hannah did then was to grab samples from various places in the in the sand recycling process, including the stalls in which the animals were housed. Took these back to the lab, would PCR amplify the 16S ribosomal RNA, sequence the products, and then use various software packages to figure out what organisms are in there. Identifying the operational taxonomic units, which is kind of a proxy for bacterial species, or at least closely related bacterial isolates. And you can use other tools to help you assess relatedness amongst these. This slide, for whatever reason, doesn't want, there we go. All right. So you can look at these data in a variety of different ways, and I'm just going to show some of this. Hannah looked at samples from the stalls themselves, from the gray water in the lagoons. That water is not gray, it is brown, but we'll use the term gray water as just a standard term for water that's being used for one purpose or another. Looked at the sand that came right off the auger, and then sand that sat around for 4 or 14 days, and took samples both from the surface of these piles as well to try to dig down a little bit to see if it made much of a difference. And this is just looking at a very high level, the top bacterial phyla. So green are proteobacteria, which are your typical gram-negative facultative organisms. The light green are bacteroides. These are gram-negative anaerobes. Excuse me, no, I misspoke. The blue is bacteroides, the gram-negative anaerobes. The light green are firmicutes. These are the gram-positive organisms. And as you can see, as you go through this process, there is a shift here, and we move to a more predominantly gram-negative population of organisms predominating in the sand piles. Now, what we don't know is whether this means these organisms are multiplying or other things are disappearing. I mean, and it could be both. We really haven't broken that down at this point in time. We don't know. But you get a greater abundance of these gram-negatives, both the facultative, predominantly the facultative anaerobes, with time as the sand sits around. And if you look at this at a finer level, looking at the top 10 genera, the pattern is similar. It's predominantly gram-negative organisms. You do see some changes in the distribution of organisms with time as well. So the scale here on the y-axis is relative abundance, with 1 being everything. And you can see with some of these samples from the stalls or the gray water, the freshly washed sand, your abundance is in the 20 to 30% range. So that means there's a lot of other things that aren't in that top 10 genera. So there's a lot of diversity, if you will, although you may not have large numbers of all those groups of organisms. And over time, then, you get a greater predominance of the your top 10 hits, if you will, of the bacterial genera. Again, this is in keeping with the idea we've got this shift to what seems to be a predominantly gram-negative population as this sand matures, if you will, in the barn area. So these were samples we collected in the summer. It was last June. And in particular, what was interesting to us, we get a predominance over time of Acinetobacter and Pseudomonas. Both of these are organisms, genera of bacteria commonly found in environment and water and soil, et cetera. And one of the things that was interesting to this farm is they have what they consider a bit of a problem with Pseudomonas in cows as they dry them off. As the cows finish milking, they've had problems with some of the animals getting mastitis and even sepsis with Pseudomonas. And so perhaps this predominance of organisms over time in the sand has something to do with that. That's something they're looking at in terms of their operation. I think I'll skip that over. You can analyze these microbiome data every way imaginable, and I won't go through much of that because if you don't work in the field, it wouldn't mean too much. But I think this graph here is useful, and it demonstrates dissimilarity amongst different types of isolates. So I've got different colors here for different samples that were worked up. So freestall, gray water, fresh sand, and then we've got those that were recovered 4 days or 14 days. And what this does show is there is a relationship here, a relatedness with time in the process. So the samples do cluster together in the summer in terms of the types of organisms you find, and this does change over time, and these changes are consistent. And it has to do with more related to time within the maturation of the sand pile, if you will, not whether you're on the top of the pile or further down in the pile. We really didn't see a relationship there. So that was of interest to us as well. We also have some samples from the winter. She took some samples last January, and what's interesting here is the distribution of top organisms does differ. Now we have a greater number of gram-positive genera that are showing up, where it was predominantly gram-negative for the summer samples. But we still have quite a bit of Pseudomonas and Psychrobacter, which are gram-negatives. But we've got Enterococcus, Aerococcus, Cyanobacteria. These are gram-positive organisms. So we're seeing a different distribution of genera in the winter than samples than what we saw in the summer. And we also don't see that same relationship to location within the recycling process that we saw in the summer samples. So there does seem to be some seasonal effect in this regard. So I should mention, we didn't find, with these methods, we did not find Salmonella as identified as one of the organisms present. Now, you can get Salmonella out of these samples if you work hard and do enrichment, et cetera, but it doesn't show up as being a major population in the sand. But we do know there have been some episodes of Salmonella in Wisconsin dairy herds that have been attributed to sand. So we decided to take a look at how Salmonella would do if you inoculate it into bedding sand. And we did first the simple experiments where you just take sterile bedding sand, sand that's not been used for housing animals and has been autoclaved, so there's no other microbes in there. Add salmonella to it, and then for a period of time, grab samples and see what happens. And what you find is not surprising if you're familiar with salmonella. The organism will persist very well for a prolonged period of time. These data go out to 4 weeks, but we've run some samples out to 70 days with Salmonella Dublin, and it dropped off about 1 log in terms of colony forming units, which is not all that much. So Salmonella is perfectly happy to persist in sand if it's not competing with other microbes. And in fact, what was also surprising to us, you actually get a little bit of growth of the organism sometimes during the first few days. So somehow there's enough nutrients, moisture, et cetera, in the sand that Salmonella can increase in number to a small extent and then persist quite happily. So that's fine. We're not asked, Salmonella doesn't have to compete with anybody in those circumstances. What would happen if you inoculated into sand that has the complex microflora that I just showed you? And this is a scanning micrograph from one of these sand samples, and we've got all sorts of organisms in here, rods, cocci, some of these a little larger, maybe they're yeast. We got what looks like a matrix here. We probably have biofilms on these sand crystals. So this is going to be a tough neighborhood for the Salmonella. They're going to have to work pretty hard to get a niche and to establish themselves. So we ran the same kind of experiments we did with the sterile sand, except here we'll use selective differential media to pull out the Salmonella. And what we find is a little bit different, but not terribly so from the data in the sterile sand. The bars in red indicate the numbers of Salmonella over time. We only went out to about 7 days for these experiments. And we do get some decrease here, approaching a 1 log decrease. But again, the Salmonella is pretty good at surviving this environment, even though there's a lot of competitors present. And this was representative of what we saw with sand from any place along the recycling process. It really didn't seem to matter too much. We do see one exception to this pattern, and that's if you inoculate the Salmonella into this gray water. And there what we find is it will actually increase in number during the same time period. So in that aqueous environment, the Salmonella does a little better, could increase somewhat in number, and might then, if this happened on the farm, could then contaminate the downstream sand that this process is putting together. So I'm going to shift gears entirely now. Then that's the work that Hannah's been doing, looking at the bacteriome, the bacterial community in bedding sand in the dairy environment, and then asking questions related to Salmonella and how it might survive. The other project we've been involved in the lab is work being done by Kurti Wadhawan, and this work has been a collaboration with Scott Rankin, who's chair of the food science department on campus, and again with Garrett Suin. And the questions we're asking here are really quite different, and that's what is it that's present on wooden boards used for aging cheese? Now this was precipitated in part because every year we have a summer program with FRI, and we take the students on field trips, and every year, and we just did this last week, we take them to the only Limburger plant in North America. And they age their cheese on wooden boards, which have been around forever. I mean, literally for decades. And I don't know if you know much about smear ripened cheese, but it's quite an interesting process. And I looked at these boards and said, I got to know what's on a board like this. Now, we don't get boards from them, but Scott did manage to get us boards from other cheesemakers who make a variety of cheeses, or facilities are doing cheese aging. There's people actually have caves or things like that where you can put your cheese in and age, and it's the idea of the environment, and the microbial environment is what's necessary to get you the cheese you want. And indeed, that's true. This particular Limburger plant, they once tried to get rid of their wood boards, and they couldn't make a Limburger anymore. They had to find some old boards and start all over again to initiate the process. So this is part of the art, if you will, of making a lot of cheeses. And that's pretty much what reiterates what I just said, in that wood is commonly used for a lot of cheese aging and ripening. Well, that's fine, but then in 2014, a crisis arose, and it happened first in New York State, where the FDA said, well, you can't use wooden boards because these are porous and microbes get in, you can't disinfect them properly. And so Wisconsin, they didn't take this too well. We have a lot of cheesemakers in Wisconsin. At one point, we actually considered making the slogan on the Wisconsin license plate, "Eat cheese or die." Unfortunately, we didn't choose that one. So everybody got agitated very quickly, and this was a national thing, not just Wisconsin, but the politicians in Wisconsin were quite vigorous in coming to the assistance of the industry, and this was reversed, I think, within about a week or so. But for a while there, it really put a panic into the industry. And although it seems intuitive that wood is not a good idea, there's actually data to suggest that's probably not necessarily the case. And there's been a number of studies done over the years, but the initial work that really raised this question was done by Dean Cliver, who was at the Food Research Institute at the time. And there have been others who have looked at this too, that they've compared wooden cutting boards, plastic cutting boards, different sorts of things. And actually, you're more likely, in some of these cases at least, and you got to put some caveats on this, to recover pathogens inoculated on one of these boards if it's a plastic board than a wooden board. Now, that would seem counterintuitive. But what Dean and others showed is that some of these organisms that go into the wood don't come back out. And wood also does have various compounds that it produces. I mean, there's certain chemicals in wood that have antimicrobial activity, or at least inhibitory activity. So exactly what's going on is not clear, and this does depend on a variety of circumstances. I don't want to just say wood is magic, but there is reason to believe that wood does provide a surface that is useful for this sort of thing and doesn't pose quite the risk you might think. So what Curdy did was to get a number of boards. Scott Rankin got them for us, and she collected samples from the surface of the board and then also drilled down into the wood to see if this really made much of a difference. And I'll cut to the chase and say the differences we saw there were not very great. It seemed to be pretty similar. Removed these samples, extracted the DNA, did the same sort of analysis I just talked about with the sand samples. And this is basically a survey at this point in time. We really haven't taken it too much further, but you do get a diverse array of bacterial communities on these boards. And these boards are different types of boards, come from different locations. A and B come from the same facility. Both places made a smear-ripened cheese. I think C might have been a smear-ripened cheese as well. And so here you've got a lot of organisms in the genus Brevibacterium, and this is one of the bugs you'd expect to be present on a semi-soft smear-ripened cheese. Get more gram-positives on some of the other types of cheese. So again, this will vary depending on what type of cheese you're making, the circumstances, et cetera. But one of the questions we're interested in asking is, okay, you have this complex microbial community on a board. What if a pathogen like Listeria comes along and lands on that board? Could it be this community is actually beneficial in terms of restricting the survival or growth of that organism? And there is some data out there, primarily from France, where people have done some studies of that sort and suggested that is the case. So we hope to go in that direction, at least to get some investigation of whether that is occurring. So with that, I'll just kind of summarize. Been involved here, described a couple of studies here looking at the bacterial species in a variety of environmental situations where you might be concerned about Salmonella and Listeria. Bedding sand has a complex microflora, and this does vary from what organisms are in the greatest predominance, does vary with seasons. And if Salmonella gets into sand, it's pretty good at persisting. That's less so if it has to compete with other microorganisms, but even in that situation, it's able to persist for a substantial period of time. And then we're going to be pursuing further description of the bacterial community on wooden boards used for aging cheese and trying to get some assessment as to what influences might have on any pathogens that might land on that surface. So with that, I'll bring my presentation to a close. I'd be happy to answer any questions you might have. Oh, I should acknowledge the folks who did the work, obviously. Hannah and Curdy, Garrett, others in the labs who helped us with these samples, and then funding from the Wisconsin Agricultural Experiment Station, the Rankin Dow Laboratory for Food Safety, and the Food Research Institute. Thank you very much.